Picture this. It's 1977, and you're standing in a dimly lit lab in Cambridge. The air has that faint, sharp smell of chemicals. Down on a glowing light box sits a thin pane of glass holding a gel, and embedded inside, dark, radioactive bands stocked up like the rungs of a microscopic ladder. A scientist named Frederick Sanger is literally tracing his finger up from the bottom of that gel, reading off a sequence, A, T, G, C. He's not just looking at a chemical reaction, he is reading the fundamental source code of life, base by base, for the very first time in human history.
Welcome to This Explainer. Today, we're diving into Sanger sequencing, the molecular blueprint. We're going to break down the elegant Nobel Prize-winning methodology that literally cracked open the genome. You know, for over 40 years, this technique was the absolute gold standard. It's what made the Human Genome Project possible. And yeah, sure, today we've got these massive next-generation sequencers, but the fundamental brilliance of Sanger's method, it's still one of the greatest leaps in all of biology. So let's get right into how it actually works. All right, we need to zoom way into the invisible molecular level. Think about a normal building block of DNA, a regular nucleotide. It has this crucial little tag attached to it called a 3' hydroxyl group. You can kind of think of it like the coupling hook on the back of a train car. You absolutely need it to attach the next car in the chain. But Sanger, he had a genius idea. He created a modified version that is entirely missing that hook. If you slip one of these modified versions into a growing DNA chain, the enzyme building it tries to attach the next piece, finds nothing to grab onto, and just stops, dead in its tracks. It is the ultimate chemical stop sign.
So how do we use that chemical stop sign? It all comes together in a cycle called the chain termination principle. First, we take the DNA we want to read, heat it up to pull the two strands apart, and then cool it down just enough to attach a starting block, a primer. Then, a heat-resistant enzyme, usually Taq polymerase, takes off sprinting down the strand, matching A to T and G to C to build the new chain. It's grabbing normal nucleotides left and right to build, but every so often, by total random chance, it grabs one of our modified, fluorescently tagged stop signs. The second that happens, bam, synthesis terminates. That specific molecule is finished. Now, what happens when you do this across millions of identical DNA strands all at the exact same time? Well, because that termination is completely random, you end up creating this massive nested set of DNA fragments of every single possible length. One fragment stops at base 12 with an A, the next one stops at base 13 with a T. By running 4 separate reactions, or by tagging each of the 4 bases with a different color, you build a vast physical ladder. And the brilliance of it is that the length of the fragment tells you exactly where that specific base sits in the overall sequence. Of course, doing this by hand on flat gels was exhausting. So to scale this up into an automated marvel, scientists introduced capillary electrophoresis. Imagine a microscopic glass tube that is literally thinner than a human hair. We inject that mixture of millions of DNA fragments into one end, hit it with an electrical current, and watch them race. The shorter, lighter fragments zip through super fast, while the longer, heavier ones lag behind. And as they cross the finish line in perfect sequential size order, a laser beam zaps the fluorescent tags, and a digital camera captures that specific flash of colored light. And that digital camera gives us a real-time chromatogram. Those racing peaks you're seeing, each one is a cluster of DNA fragments of the exact same length passing the laser. Because they were separated perfectly by size, we're reading the sequence right in order, from the shortest fragment to the longest. And check out the color coding. Green peaks are adenine, red or thymine, black or guanine, and blue or cytosine. By just tracking the colors peak by peak, the original DNA sequence completely reveals itself. Now here is a beautifully clean chromatogram read. This is exactly what every molecular biologist hopes for. The peaks are incredibly distinct, perfectly spaced, and there's almost zero background noise. A single, really well-run automated reaction can reliably give you between 500 to 800 base pairs of continuous sequence. Now, the very beginning, right after the primer, can be a little messy, and once you push past that 800 base mark, the peaks tend to widen out and get blurry. But in that sweet spot in the middle, the data is just exceptionally pristine. But here's the catch. How do we take those pretty flashes of light and turn them into highly reliable, objective data? Scientists used to have to essentially guess when a read got messy, which was tedious and, honestly, pretty error-prone. Enter the absolute hero of sequence analysis, the PHRD algorithm. PHRD is a software that mathematically rescues the entire process. It analyzes the exact shape, the spacing, and the intensity of those peaks, and it gives every single base a strict quality score. It even trims off the messy ends for us.
If PHRD assigns a base a Q20 score, that practically guarantees a 99% accuracy rate. It completely removed human error and changed high-throughput sequencing forever.
So you might be wondering, with next-generation sequencing everywhere today, is Sanger sequencing obsolete? Absolutely not. It is highly active in labs right now. Think of it this way. Next-gen sequencing is like using a satellite to map an entire continent. But Sanger sequencing? That's a high-powered sniper rifle. If you just engineered a specific recombinant DNA plasmid and need to verify the exact clone, or you amplified a gene with PCR and need to check for one single mutation, you don't sequence the whole genome, you use Sanger. It's fast, targeted, and absolutely remains the gold standard for routine verification. From 1977 right up to today, the technology has evolved from radioactive glass plates to sleek laser-powered robots. The analysis went from a human finger to advanced algorithms. But the core chemical idea, removing one single oxygen and hydrogen atom to selectively stop a DNA chain, that hasn't changed one bit. It's a method of extraordinary precision that still drives our understanding of biology. So the next time you hear about a newly discovered mutation or a verified biological clone, ask yourself, what invisible molecular stop signs were used to read that code? Thanks for checking out this explainer today, and keep exploring.